hopefully know me. I'm Debbie. It's me who sends your emails and facilitates the network. If you could put your yourselves on mute for now, that would be great. It just means that it doesn't interfere with the noise. But if you have a burning question, you can ask it or put it into the chat box, and we'll Elizabeth or Hilary will answer it at the end. But if it's a burning question that you need an answer to right then it's okay for you to ask it. The session will be recorded, Sarah, is that right? And it will go onto the Knowledge Hub afterwards. I think you're all aware that the sessions are 100% funded, as is any one-to-one advice and support. So if there's anything that comes off this session that you think you want to know more about, but on an individual basis, then please do get in touch and we can set that up for you with the right people. I'm not going to labour the points. I think you all know really actually the ins and outs of being on a Zoom call. So without further ado, I'm going to introduce Elizabeth Stockdale and Hilary Papworth. Elizabeth leads the Soil Biology and Soil Health Partnership funded by AHDB and BBRO, and Hilary's leading a project on nutrient management, so they're perfectly placed to run this session for us on soil health in flowers. So I'm going to pass over now to Elizabeth and Hilary. Thanks very much.
Thank you. It's mostly me. And a little bit of Hilary, so I apologise for that balance because it might not be quite right, but it's kind of where we currently are in terms of the information we have to share with you. So it also gives you an opportunity to say, oh, we're sick of you, and invite Hilary back for some more detail later on. So focusing on soils, on soil health, I think the most important thing to say is there is no such thing as soil in a singular. All pieces of land are unique. Although we can talk about soil types in lots of different ways, and those do group soils together in the same way as a variety groups a plant or a species, or a breed groups an animal, actually the soil is formed in its particular location as a result of that interaction between the underlying rock and the overlying climate, and then that biological interaction driving everything in the middle. And so there's some lovely pictures. I'm not lucky enough to own the book Kubiena's Soils of Europe, but as he began to describe soils and talk about them scientifically, he decided that the best way to do that was to capture these sorts of pictures, and they are truly beautiful, linking that underground geology through to the above ground community. And of course each soil is uniquely adapted to the climax vegetation community it grows. And then there's us, who over thousands of years, about 10,000 years or so in the UK, have come along and cleared that climax vegetation and put those soils to work for us. And so our soil type sets the inherent limits that there are to production, together with our climate, our slope, and things like availability of irrigation. And so we need to know about our soil's character. That is that soil type. It might be the depth, it might be the stones, it might be the texture. If you talk to a soil scientist, they can go into long, complicated explanations. I can do it, the best of them, talk about 3YR, LG, Munsell colour charts, and infiltration rates, and describe soil structures in terms of this or that kind of PEDs or whatever else. But the key is that the people using the soil have the kind of tools they need to know and to measure their soil's character. And that's usually how it responds to management. You know, the way we judge other people's characters is to ask them a few questions and see how they respond. I'm terrible if I haven't had my cup of tea, and then the guys saw me just making sure I got a cup of tea between meetings before I talk to you. Otherwise you never know how grumpy I might have been. So that knowing my character and being able to measure it and take it into account is really critically important. But we know too that management modifies the properties of those soils. So the structure, the way the particles are arranged, the nutrients they contain, things like the biological activity that might be indicated by earthworms or other things. It's important to recognise that it's really important to measure and know about those things too. And that's the part of soils that we now talk about and call soil health. And it's that information that allows us then to bring together the information about the character and the health of the soil, and then to make informed decisions about, well, what to grow and how to grow things that we're trying to, whether flowers or pick your own veg or anything else in between. And there's some very simple principles that the Soil Biology and Soil Health Project have brought together. And this is, there's a document, and I think I managed to put a link into it at the end, called The Principles of Soil Management that AHDB produced about now 9 months ago. That really tries to just bring kind of a very simple soil science textbook to put together in one place, recognizing this importance of the interactions between the physical, the chemical, and the biological properties of the soil. So here, if we think about the physical properties of the soil, that's how it works and handles and feels. And so the texture of the soil, the amount of sand and silt and clay, helps to determine how that soil will respond to the amount of water it's getting. And you in Wales know all about soils getting plenty of water. So understanding what the limits are to the workability of the soil, when it can be touched and maintain its structure, and actually when just touching it creates a smeary mess. And there's often a narrow boundary for each soil between those zones of workability, or friability, as we call it. And so understanding that is very much about knowing my soil in my place. But it's also important to understand if we can optimize that water balance, keep pores in the soil full of water when we need them, the small pores holding on to water to keep plants growing. Because just even in Wales, there are times when it doesn't rain for more than 2 days in a row, and the plants need to draw on the store of water in the soil. But at the same time, we need pores full of air so that the plant roots can breathe. And so that balance of drainage is really important to get the aerobic, anaerobic balance, the balance of air and water in the soil about right. And that means this time of year is a really good time to go and look and see how water's sitting and standing on the soil, to do things like check if drains are running like we expect them to, and to do those really fiddly and annoying jobs like cleaning ditches if we need to, or jetting drains to make things possible. And that can be at quite small scales, but can be really important in terms of holding water in a field when we don't expect it to. It just can be a small blockage in a ditch can cause a real big problem going backwards, even 2 or 3 fields back. So we might need to trace our drainage down through into our neighbours. And sometimes that means some complicated conversations, I'm aware. And how we work and handle the soil is important to minimise compaction, to optimise soil structure, to keep that balance right. So that's physical, how the soil's working and handling. If we were thinking about nutrients, you'd be expecting me to talk about chemistry, and chemistry does matter. Chemistry is determined in the soil largely by the underlying geology, giving us lime, perhaps if we're over limestone, or breaking down to give us sand over sandstone, and granite giving us that acid balance of nutrients. Basalts, there are a few in Wales giving us that much more balance. And then there's that glacial smear over the top of most things, that clay till that's a mixture of clay and rock smeared over the underlying rock. And that chemistry of that underlying materials is what our native or natural character of the soil will be. But we are able to work with that and maintain. And for different crops, it will be different. And particularly for different flower and shrub species, that optimum pH is key. And there will be some soils where actually, I'm thinking, and I can never remember the name of the plant that gives me different coloured flowers if I'm on an alkaline or an acid soil. Someone will remind me later and say, you really should have known that. And I should. It's really important there to understand what we're targeting, because we can change so many things happening inside the plant as a result of something as simple as the pH of the soil. Not easy to change if we inherit a high pH, though possible at a small scale in pots or containers, but at field scale, really, our best option is if we've got acid soil to be able to bring in lime to get that pH to the level we need. And we need to constantly keep an eye on that, because that's also determining how well the soil can function in terms of the rest of the nutrition. Because there is a beautiful sweet spot for nutrient availability around about pH 6 to 7. So we tend to talk about 6.5 being optimum for nutrient availability. It's a bit broader than that because don't forget this is a log scale for pH. So actually 6 to 7 is not just a change of one thing. It's 10 times different in acidity between pH 7 moving to pH 6. But that optimum range keeps actually the biology also in balance. So if we're in a system that requires us for our particular flower species, our other crops, to keep the pH somewhere outside that balance, we'll be making, well, the nutrients and the biology a little bit uncomfortable as a result. And so our management of nutrients will just naturally be a bit trickier. The key for getting good growth, of course, is getting the right amount of nutrients at the right amount at the right time. So it's important to know our soil textures and what our soils can supply. So getting that, understanding the capacity of our soils to hold on to the nutrients we add is also important. And for those of you who've got Zoom mastered, so you're watching me wave my hands about far too much as I talk, actually it's really useful to think about your hands in the context of buffer capacity. Because a sandy soil or a silty soil is dominated by little rocks. Sand and silt is just little rocks. And so those little rocks don't have very much active surface area. They are just little bits of gravel, but very small. If we go into a clay mineral, so we've got clay in our soils, that's formed at the Earth's surface sometime in the last some millions of years. But it's a layered mineral, lots of layers stacked together. I used to be a lecturer at a university, and I think of a clay mineral looking like that pile of A4 sheets I used to carry around between lectures. And when I was running from one place to another, they were never quite all stacked together. So that loose pile of leaves is what a clay mineral looks like, but really tiny. But what that means is it's got quite a lot of active surface area. So our sand and silt, almost none, our clay, a little bit, a good amount of buffering capacity. Now, the magic thing for buffering capacity is actually organic matter. So if people see my hands, we've now got nearly 10 times as much buffering capacity in an organic matter particle, the same weight of organic matter, as we have in a sand or a silt. So for the same weight added, we get lots more ability to hold on to the nutrients we need. So a little change in organic matter, particularly in a sandy soil, can make a really big difference to our soil's ability to hold on to the nutrients we're putting on, keep them in the right place around the plant roots. So physics and chemistry set sort of the habitat, the context, the skeleton in which plants grow. And then the biological is about that interaction between the plants and the organisms that those plants support. And I very deliberately say that because it's really important that we recognize that it's the plants, the photosynthesis of the plants, that captures and feeds all the organisms in the soil. So feeding that soil regularly is about using the plants well, diversity and different species perhaps, and organic matter inputs, bringing other photosynthesis from other places, perhaps compost, manures, to allow us to feed the soil as well as to directly feed the plant. So that photosynthetic carbon gives us the power to keep this fidget spinner that soil health going. And coupled with that, I think it is important to recognise that tillage of any sort does disrupt those webs and interactions in the soil. That isn't to say that tillage is a bad thing, because we need tillage at key points to manage and enable our plants to grow. And I've just said growing good plants is really important to feed the soil. So it's really important, though, to only move our soil when we really need to. And a really key third principle that goes along with that is this need to diversify the plants that we're growing, ideally in space and in time. So if we are looking to grow a particular crop, sunflowers perhaps, regularly, actually it's important to think about where that might sit in the field at different times. We might only have a small area, but we might need to move that crop around rather than to keep growing it in the same place time after time. Classic principles of crop rotation that you'll know from vegetable growing apply too if we're thinking about growing really good crops of anything. And in the AHDB project, we've been looking at how we can give farmers, growers the right sort of tools to be able to investigate this soil health stuff. That is to put together their physics and chemistry and biology. And the tools, and you see them in the circle there, really aren't complicated ones. A spade, somewhere to take your notes. I've got some plastic bags on my clipboard as well. That means I can remember to take a sample with me. I tend to go into the field with them already labelled, and always a bucket for carrying things around in. There's nothing clever going on in this bucket. It is just to give me something ability to carry things around in. And I will have in there too an old plastic Tupperware container, because when I dig up my soil and I'm looking at the physical structure, I also want to look at the earthworms and count them. And it's always easier just to, well, earthworms never stay still, even when you put them in a Tupperware container. But it's useful to be able to collect them together, to have a look at them and see what they're going on. So we might be used to the idea of using soil samples to look at something like nutrient management planning. So it's really useful to be able to take those mixed samples to do that. And this approach doesn't necessarily get rid of that from the system, but it complements it in the same way as most of you will be old enough, I suspect, to have been summoned by the GP. You know, when your birthday ends in a 0 and they suggest you might like to pop in for one of those regular health checks, which means they take some fairly simple measurements to give you some advice and to have a look and see if there's anything that they might need to, well, look at in a bit more detail. So this approach to looking at soil health for soils is a bit like that sort of test. It's a sort of GP's general purpose sort of soil approach. So here we're looking at going back to the same place time after time. Once about every 5 or so years will probably be enough to build us up a really good picture. If we know that this block of the field is pretty much the same and behaves the same, we'll just need one point inside there to have a look. I, at the moment now, have discovered the magic of what 3 words. I don't know if many of you have. It's a little app on your phone that actually gives you a 3 by 3 meter square on the Earth's surface with a unique 3-word identifier. It's fantastic for identifying house doors when you're in the middle of the countryside and no postcode ever works, but good too for identifying spots that you want to go back to in the field and measure. We sample only within 5 meters of that, so the little bit of wobble that a satellite has doesn't cause us a problem. And we've moved to using what 3 words because even in the middle of nowhere, and I live in Cambridgeshire, and you wouldn't think that's the middle of nowhere, but actually we have shocking mobile phone signal. And so actually it's been really useful to be able to do things without my phone really working very well and when it's offline. So we do a combination of in-field scoring with sample collection to send away for some extended analyses. And we then use that data to see if our properties are where we expect them to be. Now, I probably knew, given I knew I was talking to you today, should have put my high rainfall region slide up, because I know Wales really doesn't fall in anyone's mid-rainfall zone. But just to give you an idea that actually if we're going to think about something like organic matter in the soil, it's an area that hasn't had really good ways of interpreting that data until relatively recently. In fact, the scientific data was collected in the mid-2000s, but it hasn't been pulled together to give these sort of traffic lights. We're much more used to this sort of approach for things like nutrients or pH. But here we can see that on a light or a medium or a heavy soil, we might have to have different targets. So if we've got 3 to 4% organic matter on a light soil, a sandy soil, or a very light silty soil, somewhere around 70% sand, certainly less than 10% clay, 3 or 4% would be actually quite a good number, and certainly 4 or 5 would be more than good. But our way across there on a soil that's more like 30% clay, that organic matter level won't be sufficient. And that's, don't forget, that ability of the soil to stabilize the organic matter as well as the nutrients. So a sand and silty soil is not really very good at holding on to organic matter. And however hard we try, that organic matter will break down and turn over and not be able to be accumulated in the soil. In contrast, in a clay soil, quite a lot of the organic matter becomes stabilized and stuck. The stickiness of the organic matter becomes stuck to the clay, and the organic matter becomes quite well stabilized. And so we can have higher levels of organic matter just naturally in that soil. And so our targets are naturally higher. What does that look like when we break it out and start taking samples? So I've got here managed to get just a set of these scorecard data. These are all soils that had been used in what we would call rotations with late harvested crops. So that means things like potatoes and sugar beet, but does include, I think, 1 or 2 fields that have had both vegetables and narcissus bulbs in them through the rotation. And the physical structure here shown by the visual assessment of the soil structure is usually pretty good. But your eyes will immediately have been drawn to the red, because that's what everybody looks like when they see these things. So, not very many earthworms in these soils with those late harvested crops. But that's to do dominantly with the relatively intensive tillage that's associated here. But it doesn't tell us, or what it does tell us is that it is possible to have good earthworm numbers in those soils. And the interesting thing is not any of these scorecards on their own, but the kind of conversations that you might have if you've had your scorecards and you put them together and you say, I see you've got higher earthworms than me. What have you been up to that means you've got higher earthworms? What's been going on in your management that means you've been able to, well, I've got a sandy soil, so I'll never be able to have that. And it's those conversations really that these kind of numbers and this kind of benchmarking that it supports that's been the really strong thing going forward in relation to the use of this kind of scorecarding approach. We've also been looking at how we might use that sort of system for other systems. So I've got a set of top fruit growers working with Sainsbury's who've been working with me to think about how in their tree rows, can't draw pictures of trees on PowerPoint, I apologise. So these are crosses to be trees. In their tree rows, they've also been thinking about how to take those observations of structure and manage to record their centre. And we're currently collating that data and bringing it together. So the soil biology and soil health project's really been focused on giving those practical tools to farmers. But at the same time, we've also been trying to think about the management practices that might be able to support and improve soil health and how they might actually work in different cropping systems. So there's an awful lot of data and I'm not going to show you very much of it, but I do want to highlight for you just some of the things that we've been doing in flower crops. And this is work dominantly led by our ADAS colleagues in the team. So we had a Narcissus trial, which was looking at traditional practice for the farmer, the addition of farmyard manure, the use of green waste compost, and that combined with a mycorrhizal product. So that's a mycorrhizal fungus we find in the soil, but we've encouraged and enabled it to make a better link with the bulbs as they grow. And pretty much every crop, not every, but pretty much every crop has an ability to make this association with a set of fungi, mycorrhizal fungi in the soil. If we're growing tree crops, the mycorrhizal fungi are different. They're ectomycorrhizae and they're specific and unique, and roses too have their own specific ectomycorrhizal fungi that they need. In most field crops and herbaceous and flower crops, as well as things like cereals and root crops, it's the arbuscular mycorrhizal fungi that are important. And they make an association within the cells of the root and then reach out into the soil. They borrow the sugars from the plant directly and then reach out using that energy into the soil and are able to bring back more nutrition from the soil to the plant as a consequence. And so the question here was, would that mycorrhizal product make any difference in growing these narcissus bulbs? And we're particularly interested in seeing whether there's any interaction with some of the key diseases for the narcissus crops too. And one of the challenges is of course that fusarium can come in 2 ways. We can have fusarium in the soil potentially, or perhaps coming in the bulbs. And certainly we found a significant input of fusarium in the bulbs here when they were assessed at planting. I think was one of the things that was interesting here was actually the change in time that showed an increase in risk factors for the narcissus as the crop entered its 2nd year. This idea that crop diversity that spacing out of crops, not growing the same crop in the same place all the time, can be quite important, because what we've got here is an increase in free living nematode cysts and the lesions caused by those free living nematodes since the site was set up in 2017. But interestingly, where the mycorrhiza were applied, actually there were less damage from those free living nematodes than where it wasn't. The other thing the soil health scorecard, that general GPs thing, suggested was that we've actually got better nutrient supply where the farmyard manure was applied, but otherwise not big differences between the plots. So the incidence of those fusarium infection systems, so this is 10% of bulbs affected in the untreated situation, and we came to harvest actually some small differences between the treatments, which weren't significant, but you can start to say, well, I can see there's differences there by eye, but strictly speaking, no significant difference, no significant change here, but some indication perhaps that green waste compost was making a difference, but no difference in wilting. Interestingly, the total numbers of bulbs then harvested, so this Narcissus crop was used for cut flowers first year and then harvested as bulbs. At 85 planted originally, there was some loss of bulbs, possibly due to fusarium in the site, but interestingly, and the grower was quite excited, having thought about a way to apply this mycorrhizal product, was seeing some differences in the nosing in the bulbs where the mycorrhizal product was used. So some potential benefit of improving the ability of the plant to interact with the organisms in the soil. I was talking to someone the other day about biocontrol, and this is a sort of positive version of biocontrol, the idea that we're adding in not unusual organisms, but more of the organisms that we know might do good, and putting them in just the right place where they can make that association with the plant roots. So the emerging messages from that work, though, are that just 1 year of organic material application, it isn't really going to make a lot of difference. If you're going to commit or want to improve soil health in the long term, the approach has to be that whole rotational one. It's no good, unfortunately, I can tell you, just stopping eating biscuits for a day doesn't make a lot of difference to your weight. You've got to commit to not eating biscuits if you're going to make that kind of difference. And the same here, if we're thinking about improving our soil health, it's not just a one-hit strategy, one fix strategy. So that single year of organic matter applications on its own wasn't enough to make a difference. Though we know from other studies in different crops that a regular addition of organic material to soils tends to improve physical structure and stability, as well as adding organic material that can help with that buffering and binding of the nutrients. It's really tricky to do disease assessments in these sorts of field scale crops. And so actually, because of that variability, it's also very difficult to be clear that we're seeing a very clear difference, but we're interested in knowing and following up perhaps how some of these biological interactions can be a key strand or part of a disease management strategy within a rotation. And the practical issues of thinking about how you do soil health analysis and where we do assessments has been a really key part of this at that big commercial scale. And interestingly, even at this very big commercial scale of planting here with Narcissus bulbs, we showed that even at that scale, it was possible to do some inoculation. It isn't just a small scale thing, it can be done at large scale. That has benefits, of course, at all scales. I'm going to let Hilary just update you on her project.
Thank you for that. So I'm part of a separate project that's running for the AHDB for looking at nutrient management in protected ornamentals and in cut flowers. And the part I've chosen to talk to you about today is the Narcissus work that we're doing. So this is separate to the soil health, and this is much more about sort of conventional nutrient management. So DEFRA has the nutrient management guide, the fertilizer manual, the RB209, and this is a publication that's managed by the AHDB. The only recommendations currently within it that relate to flower and ornamental production are for bulbs, and this is part of the vegetable and bulb section. And there are recommendations for nutrient application for Narcissus. And as part of this project, we are looking at these and assessing whether there needs to be a update of the recommendations. And this sort of ties in to some of the things Elizabeth has been saying about looking at when and where you're applying your nutrient, as well as how much you're applying. So there were a number of different aspects that we wanted to look at, and the two projects really do link together because disease is one of them. But we started off the work by talking to growers and finding out their current approach to nitrogen application, and there was a real split between the different groups as to whether or not they applied nitrogen. And talking to the growers, it was very clear that over the years, because of price pressure, the rotation for growing Narcissus had really changed. Their previous approach would have been to crop Narcissus over maybe 2 or 3 years and then lift for bulb production. But a lot of the growers were saying that the price of bulbs had decreased so much that they were persisting with their cut flower crops over a large number of years. And whereas 3 years might have been quite normal at one stage, some of the people we were talking to were having, from single planting, maybe 5 years of cropping. Even some were saying that if they could maintain the viability of the crop, they were going up to 7 years from a single planting. So the nutrient of those crops, the supplementing of the nutrient is obviously becoming much more relevant because growers are keeping in the same ground. They can't just rely on the nutrient within the bulb or within the soil from the rotation. So much more interest in whether they could apply nitrogen and whether they could apply nitrogen at a more beneficial time to the crop. Now, there was a big difference in opinion on whether they were experiencing loss of nitrogen, loss of bulbs to fusarium because of the nitrogen that they were applying. So there's been quite a number of studies that have looked at this, and the AHDB Narcissus guide has some information on this, but there's been a bit of a mixed response to whether applying nitrogen to Narcissus bulbs gives you an increase in loss. And the information about the mycorrhizae is obviously very, very good, because if there is some way of mitigating this, it's an important aspect to long-term viability of these crops. So what we are looking at is, can you apply nitrogen and be beneficial to the crop? Can you increase your yield? Can you increase the duration of the crop viability? And are you having a negative impact on your field due to loss through fusarium? So it's quite a complicated picture that we're looking at. We are running the trials over 3 years, and we have sites in Lincolnshire and Cornwall. And these are 2 large-scale growers that are part of farm rotations. The Cornwall site is just outside of Truro, and the Lincolnshire one is in Spalding. So very different soil types, very different aspects to the growing sites, and very different climates. So with that, and with the difference in varieties, it's quite a complicated picture. But what we have set up in the trials is we are looking at 4 different rates of nitrogen application. So an untreated 30 kilograms per hectare, 50 and 80 kilograms per hectare application rates. And we're looking at 2 different application times. So we're trying to see if early or late application is having a difference in the growth. And over 3 years, we will be using exactly the same fields with exactly the same plots and taking assessments on flower number, stem length, and on bulb loss over those 3 years to see if there is a difference in response to our different treatments. At the end of the trial, we will be doing similar to Elizabeth's trial. We will actually be looking at the number of bulbs that are left in the plots. We know the rates that they were planted at, so we'll be looking at the final numbers, so we will be able to assess whether there's been an increase or a decrease in those areas as well. So we'll have lots of measures of how the crop is responding to the different application rates and the different timings. And we hope from this to be able to update the recommendations and better inform the growers as to management practices for their Narcissus trials. So no data as yet, but I do hope to be able to come back and talk to you later in the year when we've had a chance to assess the data that we've collected, and certainly be able to update you next year when we have our 3rd year of data. Back over to you, Elizabeth.
I wasn't trying to hurry her on, I was just being inept at my ability to operate the Zoom. One of the things we have done during the lifetime of the big project is to try and bring together everybody working in this area, advisors and farmers and academics and consultants and NGOs. Everyone seems to currently have a real big interest in soils, and it's been really important for us to try and build a sort of joint understanding of what good practice looks like and what managing soils for a sustainable future might look like. As a result, we had a bit of the kind of event you can't have at the moment. We just locked everyone in a room and didn't let them out until they'd agreed some things. And that was what would good practice look like for different kinds of crop systems. I've just picked up a couple of examples, and I'm happy to witter on about all sorts of things to do with mud when I've just shown you these. But I think there's something here, because it does, and not every flower crop's the same, but in many contexts, those practices associated with late harvested crops and practices associated with particularly large scale flower production can be similar to one another in terms of creating that added risk for soil damage quite a lot of the year, perhaps when the soil looks relatively bare and there's quite a lot of time for the rainfall to directly impact the soil. So leaving soil bare over winter means that we need to just be thinking about how we can be finding other ways, perhaps through intercropping and through understory cropping to maintain and maintain good cover. And they're in a diverse flower bed. I'm thinking of things like meadow flower establishment. Actually, that green cover can be really important in terms of protecting the underlying soil. I think part of the problem, perhaps for particularly some of the large flower growers, is the lack of control, as for people like potatoes, that we're just renting land and coming on to use land once in a point in the rotation. So there's not a lot of ability to shape or control how that land's used the rest of the time, to be able to put the land in the best possible heart for that part of the rotation. I always look at pictures of Narcissus and think they look beautiful for the flowers and also just a little bit sad for the soil all at the same time. And that putting the soil in the best possible heart in the rest of the rotation there can be really important. But that means for those difficult, potentially, conversations. And so extended rotations and just ensuring there is opportunity to recover soil health through conservation practices in other parts of the rotations can be really critical. I think looking at the whole farm as a system is important too. And I think for many of you relatively small scale growers, these things are not necessarily, you think, oh, that's not really relevant to me. I'm not doing a lot of soil moving. I'm not doing a lot of traveling on the soil. But actually, at the smaller scale, our feet and our hoes can actually have the same sorts of impact. And we need to be thinking about how we use our land and our soil in the same way, just asking ourselves in the same way as I should be asking myself about the state of my garden outside. As do I really need that much bare ground over? Could I be using some green cover? Do I really need to work the land today, even though it's a weekend and I've got an opportunity to do it? Should I be doing something slightly differently because things aren't quite working? So knowing my land and working with the principles that I showed you in the beginning, and I probably should finish back with those, but I haven't got a slide to do it. That idea that we get the physics, chemistry and biology right, and then work with our soil in its natural environment, is going to give us the best outcome. It builds us our resilience and enables us to move forward. And what that's not about is applying exactly the same practices as outlined in a book or done by my neighbour. It's very much more about that skill that's called husbandry. Good crop husbandry, good soil husbandry, well, are different than management. Well, I've tried managing my husband and I know it doesn't work. There's some different sorts of skills that are needed, some watching and thinking and responding to need. And those things are not different when we think about soils and crops and flowers. And those are those two things I said I would try and I hoped I'd had here to show you at the end. They're Principles of Soil Management, which is just a useful book, deliberately designed to work across all sectors and all types of growing systems to give us some of the clues as to what good management might look like. And also to understand why things are happening, why capping happens on this soil or not on that soil, and so on. And then a particular leaflet that we drew out relatively recently, this one, Managing and Measuring Soil Organic Matter. So a bit of more focus there on the soil organic matter, and where that leaflet is on the AHDB website, there's also an opportunity to click on and download those traffic lights for soil organic matter. So if you have those numbers, you can begin to benchmark your own soils. But thank you for listening, and I will stop sharing. And I'm happy to do, I'm a soil scientist. I trained as a soil scientist. I can talk about mud for hours. So if you want to talk about mud with me for hours, I'd be very happy to continue to do so.
Thanks, Elizabeth. If people want to ask questions, you're very welcome to either put it in the chat or to just unmute yourself and ask Elizabeth a question directly. Are you all being shy?
There is no question to ask.
He covered everything, which is amazing.
I started my presentation and went, oh, I haven't got a slide like that in, and I haven't covered that, and I should have put more of that in. So I'm sure I haven't got the balance quite right. So there will be things I've missed. It's really difficult with a group I don't know to know what you know and what you don't know. So there might be some really foundational things you don't know, or there might be some, you haven't covered that really interesting extra thing, that the next level that you want to just know more about. As I say, my background is in soils, and so most soils things I can have a fair go at in terms of thinking about how we help soils to work best for us.
And Justine has put a question actually in the chat there, Elizabeth, which is, how do you feel about no dig?
For the principle, and I'll just go back to saying, well, the principle only till when you have to. So absolutely, but I also recognise that some situations you can't. That doesn't work. You know, in the bigger scale of agriculture, rather than at the smaller scale of garden production, potatoes can't work without cultivation at the moment. We're looking at systems that are much reduced or no-till for potatoes, but the same is true of some crops at the smaller scale too, that without some cultivation, they just become overwhelmed perhaps by other more competitive plants around them. And actually then it's about managing that competition. And so I think as a principle, only till when you have to is the right one. Don't beat yourself up if your system requires you to till, to dig, but also think about whether you really need to. So does the soil really need me to put it, or does it need this digging to put it into the best possible condition for the crop establishment? Actually, if I pick it up without digging it, does it break apart just as easily as if I put a spade in? At which point, why am I bothering? I could go and have a cup of tea instead of digging the plot. It would be much more effective. I noticed too, cover crops really useful in terms of providing ground cover. So literally stopping the raindrop impact directly on a soil surface can be really good for bringing new biodiversity into the plot, depending on what crop you grow. And at their heart, they're really good because what they do is keep some photosynthesis going. If the ground would have been bare and now we're growing a cover crop, what we're doing is providing energy to that below ground system to keep it going, when otherwise it would be going, well, everyone stopped feeding me, where's everyone gone? So actually that photosynthesis, the energy being captured by the plant, is fed through the root system. Around about 20 to 40% of the photosynthesis that's captured above ground is actually just leaked out around the roots and feeds the biology in the soil to keep it going. So cover crops also have a really important role. If you've ever had a cover crop and a bit next to it that's bare and you've dug them both, you will nearly always, not absolutely always, but nearly always see more worms in and amongst the cover crop roots than you do in the bare soil. And that's that indication of that faster the biological activity kept going. And that in general, in most systems, is a good thing to keep that balanced biology. It'll certainly add, so we were talking about donkey manure and what that might do in my compost heap. What it'll certainly do is add some nutrition to actually power that compost, probably to break down more quickly than if I wasn't adding it. So I'll get a faster compost where I'm adding the nutrients in something like donkey manure. Too rich is tricky because too rich is about how you use it. So if you put on the same amount, if you had donkey manure and you didn't, you'll get a lot more nutrients that might make your plants grow really quickly and really sappy, so that just don't put on as much. So it's about how you use the material you've got, understanding the materials you've got. So it's a really important thing to not do things, but to think about what that means. The answer to all soil science questions are, it depends. So should I add donkey manure to my compost heap? Oh, actually, it depends. Well rotted cow manure versus horse manure. Yeah, so the idea that we can get the material to break down really well before we add it to the soil means that when we add it to the soil goes, well, there's some more of the stuff we're used to, because that well broken. So we don't really disturb the soil biology very much when we add down that well rotted or composted material. It's already gone through a breakdown process out of the soil. And so that means that both the nutrition and the kind of breakdown products that are there are all ones that are really familiar to the organisms. So it does them a lot of good. They don't get very excited though. So it's like feeding me a really good salad. It's all right, but it's not as good as cake, is it? I don't know anybody, but things like your horse manure are more like that stimulus of sugar and a bit of, so they're doing something slightly different. They change and they upset, but not in a bad way, the organisms. They just adjust for a little while they're able to use and turn over that material. So you wouldn't use them to get the same result. So my well-rotted cow manure, going to go into my soil, get it ticking over nicely, going to help with my structure, going to do composty stabilizing sort of things. If I want quick nutrient release, my horse muck is going to give me that activity and that drive in a slightly different way. You'd have to have them analysed to know if you've got the same amount of phosphorus or potassium in them if you're trying to use them as nutrient management materials, because I would expect them to be slightly different. So a direct horse muck is like to have more nitrogen and a little bit more fresh potash in it too. Wormers, we know they have an impact on the soil. So if they're still active wormer in some material, and that's usually if we've composted the muck, it usually gets broken down during that process. If, as long as the muck's really turned into compost, gone to 60 or so, and done the breakdown process. In fresh muck, we know, and when the data shows that fresh muck with fresh wormer in it basically just sits on the soil surface for longer. The biology that come in and use it get a bit grumpy, because the worm is designed to slow down biological activity. That's what it's there for in the horse gut, and it just keeps doing that afterwards too. So in livestock, where a worm has been used, it just slows down the breakdown of that material. It's not a bad thing, it's just a slowing down thing. So it can just make the material last a lot longer. On the surface of a soil in a pasture, it would sit for longer, but in a compost heap, it would just take a bit longer to break down, for example. Oh, Emma's asking me a really tricky question that she is correct. It's definitely an it depends question. The first question is, can you stop it turning into a swamp by stopping water coming in sideways? So there's an interesting question with if you've got a relatively small plot, can you do something with a gravel trench just to minimize the amount of water coming in? So that's tip one. Tip two, can I do anything? Oh, it's really tricky, because as any crop with good root structure grows, so things like phacelias, buckwheats, even things like rye, they can make really good root structures, and they can do a good job of moving water if it ever gets sunny, so that they will move water through. The problem is they also shade the soil surface. And so in a clay soil, you've got two processes drying the soil. One, the roots taking the water out, and one, just direct evaporation from the surface. And so actually in a clay soil with a cover crop growing in it, at 10 centimetres, it'll be drier than if I didn't have a cover crop, but in the very surface, it'll probably be wetter. And that can be a really tricky thing to manage when I then want to go and work it. So it's a really tricky one, heavy clay soils and using cover crops. Or the easy soils, most circumstances, are those ones that are called medium in the middle. Those are the ones everybody wants, but actually most of us have things that are around the edges of medium. They're just a bit annoyingly heavier, or they're just a bit siltier, which means that they slump really easily when we've cultivated them, or they're just a bit too sandy, so they don't hold on to nutrients very well. But the very heavy clay ones, actually getting roots in there, just active growing roots, is actually a really good thing. And which thing to grow just depends a little bit on what will grow well. So if it does sit wet, you need to find something that will grow really well for you, but not interfere with what you're planning to grow next, because that's the other problem, isn't it? That we don't want to create a problem with a green bridge or a move of a disease from one crop through to the next while we use a cover crop in between. I'm not the best person, you can kind of tell, to advise on exactly which species in exactly which slot, but I think packets of seed are cheap enough for us to try a few. Have a look on those websites, so those things they sell you, and say, oh, I'll try a little bit of that and a little bit of that, and buy a couple of packets and just see which ones really grow well for you. If you've got good growth above ground, it means you've also got good rooting below ground. The two go together. If you've only got a little bit of growth above ground, you'll have some roots, but actually a crop that grows well will also have good roots, and that's what you're really looking for. So it means you can use your eyes to judge which ones will be doing the best sort of thing for my soil.
Thank you, Elizabeth. Are there any more questions? I'm aware we're coming up to time. Actually, no, that's perfect timing, isn't it? We're 3 minutes off 5 o'clock, so amazing. Okay, well, I think, oh, there we go. Thanks, Emma. I think we'll call it a night. Can I just say a massive thank you from all of us, Elizabeth and Hilary. So informative, really interesting, and I'm sure that we'll all take lots from what you've said. And we'll look forward, Hilary, to hearing more about the results of some of the trials. Just to let you know as well that Sarah from Tyfu Cymru has just given me a nudge that we are launching a bioprotectant support network on the 15th of March. So if you're interested in hearing more about that, then contact Sarah DeMay or Tyfu Cymru or me, and we can set you up with the right information and the right events there. So can I say thanks very much for coming, everybody. Thanks for your time, and we'll look forward to seeing you at the next event, hopefully. Bye. Thank you. Thanks, everyone.